do today is I'm going to show you first, I'm going to demo how to inoculate plates. And this kind of goes in line with what we've been doing so far. So last class I showed you pouring agar plates. They've solidified, they've cooled to the point where they're solids. And now we're going to be using those agar plates to grow bacteria. So let me take you over to that area for our lab benches. And let's see. All right, so I'm going to just kind of remind you of some of the basics that we've been doing. All right, so here's our setup that we basically had from last class as well. Hopefully you guys can see this okay. I want to make sure I'm wearing all of my safety equipment. So I've got my lab coat, my very colorful lab coat. I just want to make sure I'm not getting bacteria and stuff on my clothing because that would be kind of difficult to clean up when you're in school. So you want to make sure you got your lab coat on. I'm also going to wear goggles, going to wear gloves because we're going to be using live bacteria. So, and we're going to be using massive amounts of live bacteria. So we want to make sure that, you know, I'm basically covering the parts that are important. Eyes for sure. We don't want to get splashes of bacteria in our eyes and I'm using the Bunsen burner. So I definitely don't want, you know, the Bunsen burner, the flames to have any sort of issue there. My hair is pulled back. I don't have any loose clothing on. I'm going to put my gloves on. And so we're going to be inoculating 2 different kinds of bacteria onto 2 different plates respectively. So 1 bacteria on one plate, 1 bacteria on another plate. These were the plates from last class. I just kind of wrapped them in saran wrap just because I didn't want them to dry out because they will dry out eventually. These are not my finest work when it comes to plates. The agar wasn't fully melted, so honestly, if you guys were in class doing this, I probably would have redone all of these, but because it's me and I'm demoing it, it will work. But not my finest work with these plates. So these are the agar plates. I stored them, you might notice I stored them upside down. And the reason for that is they were in the refrigerators because I don't want, I want to keep them, as long as possible. And so if you put them in the fridge, they actually last a little longer, and wrapped up, they last longer. So sometimes they actually get condensation on them. Lots of like moisture builds up in here. Well, that would be really bad if I have moisture building up and I have bacteria. It'll cause the bacteria to basically slosh around. I don't want the bacteria to slosh around. I want them to stay put on the agar. So you can kind of see, I'll turn it upright. So you can see that the bottom section here, the lid can go up. I don't want to open the lid too much because I don't want to let bacteria in here. But the bottom section is covered with that nutrient agar. That's the stuff our bacteria will want to grow on. And so, but it's going to like grow in place. It's not going to be sloshing around as if it's in liquid. However, if there's a bunch of moisture in here, like you can actually kind of see there's some moisture on top of the plate, some little water bubbles. That condensation is fine if it's on the top of the plate, nothing's growing there. But if it grows on the layer of the agar, that would be really bad because then our bacteria just kind of move around a little bit more. They can slosh around and we don't get really nice colonies, which is what we're going for. Now a colony is, I'll kind of just draw out what a colony is going to look like. A colony of bacteria is basically, it starts out with just a single bacteria and they're super tiny. Like you won't see a single bacteria pretty much like ever in this class. But once you get a lot of them and they go through binary fission and they replicate and replicate and replicate, you're going to eventually see them sort of form. It'll look like a little dot. So that might be one colony there started off with just one single bacteria that underwent binary fission lots of times and replicated to the point where there's a whole glob of them now. You might have another one somewhere else, another one somewhere else. Those are all different individual colonies. We want individual colonies. We want to know that if I was to scoop that little colony up, that it's just one type of bacteria. We don't want this whole plate to be full of bacteria, and then I grab a section and just kind of hope for the best and hope it's one type of bacteria. So we want to be able to isolate individual colonies of bacteria. When I say isolate, it means pick up one individual colony and not like a big whole slap of bacteria. So I'm going to show you how we can get these bacteria to grow so that they show up like individual colonies and it's not just like a whole bunch of bacteria all over the place. We're going to do what's called a streak plate. So I'll start off just inoculating. Inoculate is a fancy word for saying adding bacteria. And then we're going to do a method that's called the streak plate method. And when I add my bacteria, I'm going to not just pour it on here. I'm going to use my loop. This is a loop. It's basically a little metal rod basically that has a piece of thin metal wire, and then there's literally a loop at the end. This loop at the end is where our bacteria is going to sit. So I'll actually have a little bit of nutrient broth liquid that has a bunch of bacteria on it or in it. And then I'm going to use this little end of this little rod to transfer it to something else. So I'm going to transfer it onto this plate. Now I don't want to just sort of pour the stuff on here. I want to just like color, slop this around a whole bunch. I mean, I guess technically you can, but then we're not going to get these nice colonies. So to get nice colonies and not just a big, smear of bacteria everywhere, we're going to do what's called streak plating. So imagine there are 4 quadrants. So I'm just going to kind of draw an X on my plate. So I've got quadrant 1, 2, 3, 4, and I will actually number these. You don't have to always number them, but I'm going to. And my Sharpies, not really wanting to write because there's some condentation. So 1, 2, 3, 4. 1, 2, 3, 4. And so we're going to start out with section 1. So when I have my loop full of bacteria, I'm actually going to swab this in one direction, kind of back and forth, zigzaggy through that one quadrant. So it will kind of look like this. I don't know if you can see that. So that's going to be quadrant 1. Then I'm going to flame my loop because I want to kill the extra bacteria that's just hanging out on that little metal piece. Flame it in the fire, in the Bunsen burner. And then I want to actually drag just a little bit of bacteria from here into the next quadrant. So I will literally take that loop and I'm going to drag it through one of these rows towards the top and bring it over to the next quadrant, zigzag it back and forth here. Okay. Then I want to do the same thing. I'm going to flame my loop to get rid of the excess bacteria that's hanging out on the loop. And I'm going to drag it through the previous quadrant. My Sharpie's kind of running out. Drag it through so it kind of picks up some of those bacteria. Bring it over into the next quadrant, zigzag back and forth. Flame my loop and then kill off those bacteria. And then again, I'm going to drag it through that previous quadrant. Just once it grabs a few of those bacteria and it brings it over and I zigzag back and forth here. So this is kind of what a streak plate would look like. Obviously not with Sharpie, but this would be done with streaks of bacteria. So ideally, we would have a ton of bacteria in this first quadrant, less in the second, even less in the third, and maybe only a few in the fourth. So this third and fourth quadrant, these are probably the ones where we're going to get those individual colonies to show up, where it's just one bacteria that replicated until you get that single colony. These up here are probably just going to be smashed full of bacteria. We won't even be able to see individual colonies. So I'm going to set this up now, and then we'll see next class if it really does show up where we have these individual colonies, and kind of in the first and second quadrants will be totally a mess of bacteria. Third and fourth should have individual ones that we can isolate. Okay, we're going to use 2 different kinds of bacteria. One is E. coli. So this is E. coli. This is Escherichia. And this is nutrient agar. So this is liquid. And it looks like there's a little precipitate, a little bit of haze in there. That's the bacteria. And so we're going to be taking just a little bit of this and adding it to the plate. It's inoculating. And then the other bacteria we're going to use is called Bacillus megaterium. And we'll talk about the naming of these. So this is actually grown on an agar slant. So last class I mentioned the agar slant just briefly. So what this is there's actually agar in this test tube. When it was solidifying, they kind of tilted this tube so it would solidify on an angle. That's why we call it a slant, because it literally is slanted agar. And then they allowed that to cool. And now when you put it upright, that agar is still on that slant. And then it's hard to see, but there actually is bacteria growing on the surface of this slant. So I'm going to be able to grab a little bit of bacteria from here and then also inoculate it, put it on a new plate. Okay. Anytime we are doing bacterial work, any experiment work at all, you always want to make sure you label everything properly. So I've got 2 plates here. I have 2 kinds of bacteria. So one bacteria is going to get E. coli. I'm going to put that on there. I'm going to put the date. What is the date? 23rd. And I'm actually going to put my name. I'll just put my initials. So E. coli, this might be backwards for you guys, E. coli, the date, and then initials. And notice I put it on the bottom of the plate. The reason I did that is sometimes if you have multiple plates and you're taking the lids off because you're adding bacteria, every once in a while, well, it shouldn't happen, every once in a while the plate lids get mixed up. Well, if the lids get mixed up, then I have no idea what's actually on the agar. Because the lid and the agar can be separate from each other, right? You can take the lid off, hopefully not put it down on the counter, but sometimes it happens. And then you put on a different lid and all of a sudden you think you have E. coli, but it's not E. coli. If you put it on the bottom, that's the part that has the agar. That's not going anywhere. So I always label the bottoms so that if there's ever an issue where the lids get mixed up, at least I know that I still have my my bacteria on the agar is still the same as what the label is. All right, again, I'm gonna label Bacillus megaterium with the date on the bottom. And then I wanna make sure that on that second plate, I also make my quadrants. So again, make my kind of X, label 1, 2, 3, 4 for my different quadrants. Okay, so we're gonna be using the Bunsen burner. Remember, nothing means over. You always want to do your work in front of on the sides so that you don't accidentally burn anything. Your body should actually be sort of behind this counter, so I'm never going to do this. It doesn't take much for your hair to singe, and there has been some hair singeing in previous years, so usually you can always tell hair smells really strong. So I'm like, oh, somebody got too close to the Bunsen burner. So you always want to make sure you're not ever doing this. It doesn't take much for little bits of hair to kind of start a flame. So my Bunsen burner, I'm going to have to turn on my gas first. I want to make sure my striker is ready to go when I do the gas. So when this valve is turned in line with the tube, that means it's on. So I'm going to turn this in line. And as I do that, I have a secondary valve, a safety valve. That if that one's closed, nothing's coming out of here. All right, so let's do that again. You can hear it. And now usually you can hear it. It's actually kind of hard to see the flame. You want a nice blue flame, and sometimes you can hear the difference. Like that's a nice blue flame. That's not a very good blue flame. The blue flame will give you kind of like a triangle shape at the top, and at the very top of that triangle is the hottest part. All right, I'm gonna turn that down a little because we don't need crazy wild flame here. All right, so I've got my striker can go off to the side. I've got my loop, I've got my plates. Let's do E. coli first. So here's my E. coli. And you guys can still see this. I'm a little bit farther away. So now I want to flame my loop. I'm going to bring you guys a little closer. I want to flame my loop before, in between, and after every usage. This thing is going to actually connect directly onto the agar plate. So whatever's on here, even if it's like, I mean, literally, if I do this, I probably picked up a bunch of bacteria. I want to kill that bacteria before I touch anything. So I'm going to flame my loop. So you put it in kind of that hottest part of the flame, which is right at the tip of the blue triangle. And you actually let it kind of get a little bit glowing red hot. I don't know if you can see that from where you're at, but you kind of let it get pretty glowing, like a glowing ember. And now I have my test tube. I want to give my loop a little bit of time to cool, because if I just kind of insert this right away, it's like really hot. So it basically would singe all of our bacteria and they wouldn't be alive. So I want to give it a little bit of time near our aseptic area. So we're practicing good aseptic technique. This heat from the Bunsen burner is a high pressure. It's basically pushing the air outwards. So we're not going to get a lot of bacteria from the air flowing in because it's sort of being pushed out. So that's why we always work within this space. Now I did kind of just move my loop out of the way a little bit over here sort of. So I want to flame it again. The last video, remember, when in doubt, flame it out. I kind of like that little saying. All right, so now that's good and flamed. I'm going to open up my test tube that has my E. coli in it. And this has massive numbers of E. coli. We're talking hundreds of thousands, probably millions, in fact, in this little test tube. Now, when I take off the lid, I actually want to flame just the very top of the test tube, just briefly, in case there's any bacteria that have sort of hung onto that around the edging of the lid. I'm going to put this in the test tube. I'm going to try not to touch any sides of the tube. I'm just going to go straight into the tube and then straight out of the tube. I should actually see like a little mini almost bubble on the top of that loop. Flame the tube again, put the cap back on. Notice I didn't put the cap on the table. I'm holding it in my hand. If at all possible, you don't actually want to place anything on the lab bench. Even if it has just been cleaned, it doesn't take long for bacteria to get there. We don't want to accidentally contaminate our samples. Same thing with the plate. I don't actually want to take the lid of the plate off and put it on the counter. That's just one more way that I can contaminate things. So I'm actually going to hold the lid with one hand, or hold the whole plate with one hand and the lid open. And then I'm going to take my loop and I'm actually going to just drag it in that zigzag pattern in quadrant 1. And there we go. That's quadrant 1. I don't know if you can see quadrant 1. You probably can't tell, but there are little zigzags in there. I'm going to loop, flame my loop again because I want to kill off the bacteria, the excess bacteria that's on my loop. And now what I'm going to do is, again, I'm going to drag my loop through quadrant 1, bring some of that bacteria over into quadrant 2. My loop is cooled enough to be able to touch. Lift up my plate with my one hand, lifting up the lid. It's hard to do both. And drag. Don't do that. Drag my loop. My hand's got a little wet. I just accidentally hit my loop, so I'm going to flame it one more time. Drag my loop through that previous one and then bring it over. Flame my loop. I think I was trying to be clever and lift everything up. You actually don't need to even lift the Petri dish off the table. You just need to have the lid. I was trying to hold it up for you guys. So I won't do that because clearly my hands were slippery. All right, next one, you're going to drag it through quadrant 2, bring it over into quadrant 3. And then zigzag back and forth, flame your loop, let it cool, because again, we don't want to singe our bacteria. Give it a little bit of time. And sometimes if you want to test, like, I don't really know if it's cool enough, if you kind of test it by going to an area that there is not bacteria, just the agar, you just kind of touch it to the agar. If you hear it kind of, that means it's too hot. It's basically like a little mini boiling. So you want to give it a little more time. All right, drag it through. I'm going quadrant 3 to quadrant 4 now. Drag it through quadrant 3 just a little, and then zigzag on quadrant 4. Flame my loop again. And now this plate is done. We've got all 4 quadrants. If you see it, you probably can't see it here, but like in the light, you can kind of tell where those zigzags are. And that's kind of a good way to just check, yeah, I did all 4, I did it properly. Now we're going to do the other one, which is Bacillus megaterium. This is the one that's on the agar slant. So I'm going to be having to pick up bacteria from here, and we're inoculating this plate that does not have any bacteria on it right now. Flame my loop because I forgot if I flamed it or not. So it's always good to flame, forget. Check where my quadrant 1 is on the plate. Open the test tube. Flame the top of the test tube, just the lid. Now I'm going to try not to touch the sides of the tube as best as possible. I'm going to drag it up, pick up some of that bacteria that's in this vial. Flame the top of the tube again. Close it up. And then I'm going to be lifting the lid and then adding this to quadrant 1. Remember, you don't flame after you've already added your bacteria. It will kill the bacteria. Added the bacteria to the plate. Now I can flame because I want to get rid of that excess bacteria. Okay, let it cool for a little bit. Bring it over. Bring a little bit from quadrant 1 into quadrant 2. Just a little bit of the bacteria. You basically just pass it right through one of the rows, one of your little zigzags, and bring it over to quadrant 2. Flame, get rid of that excess bacteria. Let it cool. I'm going to touch a little side of the agar just to make sure that my loop is nice and, that one was too hot. So I want to let it cool a little longer. Again, if you hear like it's, when you touch the agar, it means it's too hot. You just killed whatever bacteria is on there is already dead. So it means you kind of got to go back to the previous quadrant. All right, so I'm dragging from quadrant 2, and 2 or 3. Quadrant 3, that was quadrant 3. Flame the loop, and now I just have one more quadrant left. So So the last one is hopefully the quadrant. Quadrant 4 is hopefully the one where we're going to get individual colonies and not a big massive amount of them. Just make sure that's quadrant 4. Okay, drag it through quadrant 3, bring a little bit of bacteria over and zigzag through quadrant 4. Flame my loop, get rid of that extra bacteria. And now I'm just going to put this on the counter because I killed off the bacteria that's on it. I will wipe down this entire counter when we're fully done, but I'm not using this again right now. So now my 2 plates are ready to go. I'm going to turn off the Bunsen burner. My tubes of bacteria are closed. Everything's closed up. I don't want to blow this out like a birthday candle because gas is flowing through it no matter if it's lit or not. So I want to turn it off by just shutting off the valve. And you always want to make sure the valve is really closed, so kind of double check that the valve is really well closed. So now I have my 2 agar plates. Agar right now is on the bottom, bacteria is sitting on the agar. When I put these in the incubator, I want to flip them over so that the agar is actually at the top of the plate and just the plate lid is on the bottom. I know that seems a little bit weird, but the idea is we don't want condensation to build up and land on our agar. And basically all of our nice streak plates that we've just done, that bacteria gets sloshed around because there's just water in here now and it's just kind of like flowing around. All right, let me take off my one glove so I can use my computer. So it's possible, I mean, I was pretty careful, but it's possible that like my gloves actually have bacteria on them. So I don't want to go touching my computer right now with my gloves. I actually want to remove my gloves and then wash my hands after I've returned all these things. But I'm going to grab my computer with my non-gloved hand. Typically you wouldn't multitask like this. And we're going to take these Petri dishes and we're going to put them in an incubator. So this is an incubator. Basically, it allows you to hold a really standard temperature. We're going to be using, I think we'll do 37 degrees with this. Sometimes you can do 27 degrees. And they're just basically going to sit in here at that temperature for the next couple days. And we'll take a look next class at how they develop. All right, now I can take off my gloves. I actually would have preferred to do a glove and glove method or the beak method. And I will return the rest of these supplies in just a bit. All right, you always want to, since we're practicing good technique, after you do bacterial work, you want to make sure you wash your hands, even if you've been wearing gloves. Ideally you're doing this for your 20 seconds, which is the amount of time you should do for COVID and stuff too. Do your happy birthday twice. All right, I'm kind of zipping through here. It's probably not a full 20 seconds. Do as I say, not as it is. All right, and so now I can basically done. I should have removed my coat also. All right, don't do this in my order. All right. Okay, so that's basically how we inoculate a plate using cultured bacteria. And so we should, next class, have hopefully clearly defined colonies, isolated colonies, and we should have them for E. coli and then Bacillus megaterium. And so next class, I'll kind of show you the update for that. All right. So when we get back on Tuesday and look at some of those plates, we're going to do some additional techniques with them. So additional experiments. It's not just did they grow. I mean, yes, that's important. And we want to know, did they grow? And we want to look at the